Okay, so let's get started. Welcome everyone to our today's museum seminar. So we have a treat today. So I'm absolutely thrilled to have Ling-Ling on our platform. I think a lot of us probably already aware of her remarkable work. It's a little bit different from our conventional museum seminar because I think we are going to learn a lot of RNA biology today. So very, very brief introduction about Ling-Ling. So Ling-Ling currently is a principal investigator in Center for Excellence in Molecular Cell Science, Chinese Academy of Sciences. So Ling-Ling actually obtained her bachelor degree in Lanzhou University in biology, and then continue her master's study in pharmacology in Shanghai Institute of Material Medica, Chinese Academy of Science. After which, she actually have a quite interesting career. She pursued a MBA degree in UConn Business School in Connecticut. Which I think that's probably one of the reasons why she's so successful. I think we probably all should learn a little bit of management about the lab before becoming PI. Afterwards, she continued to pursue her PhD training in biomedical science, also in UConn, and then did a postdoc also in molecular biology in UConn. So from 2011, Ling-Ling started her from 2010, actually, Ling-Ling started her independent research program in Department of Genetics and Genome Science in UConn's Health. And then shortly afterwards, she moved back to China as a principal investigator in Shanghai Institute of Biomedicine, Biochemistry and Cell Biology, Chinese Academy of Science in Shanghai. So from 2017, she became associate director in State Key Laboratory of Molecular Biology. And then from 2022, she became director of Key Laboratory of RNA Science and Engineer in Chinese Academy of Science. As I just mentioned, Ling-Ling's work is really groundbreaking in RNA biology. Her research mainly focuses on non-coding RNA, a giant and a virus class of RNA molecules. Her group has pioneered method for studying of non-polyadenylate RNAs and contribute to discovery of the widespread expressed circular RNA and siRNA related linker RNA. Her group has characterized their biogenesis pathway, uncovered their essential roles in modulating nuclear condensates and in regulating the innate immunity, and recently linked their dysregulation in rare and autoimmune diseases. Her group now continue efforts on elucidating the biogenesis and function of these regulatory RNAs in different cellular contexts and in relevant human disease. She's actually, I think her work really quite remarkable and very, very productive. A lot of work was open new field in RNA biology. So because of these quite amazing achievements, she was awarded with numerous awards and honors. It's really long list. I'm not going to name all of them. Some of them, for example, like the Aurea China for Women in Science, Tan Jiazhen Life Science Awards. She was also selected at Howard Hughes Medical Institute International Research Scholar. And just this year, she was selected as a new cornerstone investigator, which is a very prestigious award in China. So I think without further ado, we are really, really happy you're on our platform, Ling-Ling. Very much looking forward to your talk. Please go ahead.
Oh, wow, thank you, Chen. Pleasure to be here. It's very generous introduction. So my lab focuses on RNA study. So I will spend maybe 2/3 of my talk focus on circular RNA and spend the rest of my time on the finding using the cell biology approach to study RNA in the subcellular regions. So let's see. So quite a while ago, the application of RNA sequencing has uncovered the pervasive transcription of the genome, including many long non-coding RNAs. However, at the time, those long non-coding RNAs were also thought as mRNAs. They have the polytails. So very often when people study the transcriptome, they basically focus on the oligo-DT enriched transcriptomes. So when I started my lab, I wanted to study some new RNA species. So the first experiment we have done is to look at the non-poly RNA transcriptomes. So this is the data. This is the first experiment we've done. We basically get rid of all the polyethylene and also depleted ribosomal RNAs, and then did the RNA sequencing, focusing on the long RNAs. So these long non-poly RNA sequencing data sets have identified many interesting signals. Those came from the intron regions, and also those non-poly signals came from exon regions. So those were quite unexpected because we know introns are rapidly degraded after splicing, and exons normally came with those polytails. So what are these signals? So inspired by this very early observation, we focused on this non-poly RNA sequencing data sets for a while. In the beginning of my lab, the first 4 years, we basically wanted to understand what mechanism can keep these intronic and exonic signals from degradation. It turned out that if the intron has those SNO RNA at each end that can protect the intronic sequences from degradation, forming the SNO link RNAs. Some of the introns have the C-rich motif that close to the debranching size. So those introns can be inefficiently debranched and forming these Larry intron-derived circular RNAs. Well, another circular RNA, which are even more predominantly identified by several other labs as well between 2012 and 2014, those are backsplat circular RNAs, which is also going to be my focus today. Later on, we found the polycistronic transcripts-derived SPA RNAs, whose 5' end is SNO RNA, while 3' end is poly A tail. So all of these RNAs, they do not have their own promoters, but are rather processed from those precursors of POL2 transcripts, and then stabilized by distinct mechanisms. So the question became, what do they do? So over the last 10 years, we've been focused on one is circular, another one is, as I have mentioned, the cell biology derived tools to study RNA process and function in nuclear condenses, because all of these RNAs we identified are very weird ones, not like the canonical ones. Once they produce, they tend to retain in the nucleus for these linear RNAs. We normally use cultured cell lines, and recently we all started to work with animal models. Well, circular RNAs. So circular RNAs were originally identified in 1976. This is a beautiful EM photo. We captured the circular viral RNA from the plants. And then in the 1980s, many circular RNA intermediates have been identified from non-coding sequences, most of those from introns of archaea ribosomal RNA precursors or tRNA precursors and so on. So now those catalytic introns have been well adapted for in vitro synthesis of circular RNAs. And the third group of circular RNAs we define as spliceosome dependent. Those two groups of circular RNAs that I have just mentioned, intron-derived and exon-derived circular RNAs. And for this type of RNA, the very first RNA of this type was observed in 1979, but with unknown origin and function, followed by the identification of a handful of cases of them. So now we know they're widely expressed in mammals and plants, often in the tissue and cell-specific pattern. So here shows the typical genes contains multiple exons. Kinetic splicing ligases all the exons together to form the linear RNAs, while back splicing catalyzes the distal splicing sites to form the circular RNAs. So the question was, how the spliceosome can overcome this spatially unfavorable reaction. We found that very often those circular radical exons are flanking by a pair of ALU elements, which are very abundant in primates, accounting for more than 11% of our genome. So they can form this transient pairing that bring distal splicing sites to close proximity to form the backspliced exons. So I wanted to acknowledge my computational collaborator, Li Yang. So he moved his lab from CAS system to Fudan University last year. So his lab has developed many different pipelines allow us to reliably identify this different type of circular RNAs. So the containing of these ALU elements is not sufficient for circular RNA formation because it really depends on where this RNA pairing is formed. If this RNA pairing forms within individual intron, it just has this canonical RNA splicing. Only if the RNA pairing occurs across the flanking intron of this the future circular RNA exons, now that can induce this exon backsplicing. We have confirmed this finding with a very simple experiment. So let's look at the number 2 plasmid. So these 2 blue exons can form the circular RNAs that's flanking by a pair of circular RNA. Let me have the laser point. As shown in the northern blotting, that's just strikingly circular RNA induction. Introducing the competition as shown in the vector 3 can diminish the circular RNA formation. So this is also the plasmid now we use to do the knock-in circular animal. So keep in mind about this plasmid setting. All right, so because of the widely distributed ALU elements and competition of RNA pairing in human introns, we observed more than 50% of the expressed genes, for example, in H9 cells, can produce more than 1 circular RNAs, which is phenomenally termed as alternative circularization. So this shows a famous gene called DAMT3B. Computationally, more than 10 circular RNAs can be identified over here. And those highly expressed circular RNAs are readily detected by northern blotting. And the formation of alternative circularization is not random. We have identified those 6 different types of alternative circularization events. So basically, those are 2 alternative backsplicing events, have the 5' backsplice site selection or 3' backsplice site selection. For the circular have the same backsplice site, we identify those all 4 types of canonical splicing events can be found within the sequence, including intron retention. For example, this intron can be retained in the final sequence or excluded from the final sequence or cassette exon events, as well as alternative 5' and 3' splicing site selection. So it sounds very boring, but it's actually very important because with time, people have studied this functional circular RNAs. So the current annotation of the eukaryotic circular RNA basically following the rule with the preface of circ, followed by the gene's name. However, it cannot discriminate your circular RNA from my circular RNA for those that can produce more than 1 circular RNA from 1 locus. So we encourage in the field that to include the particular exons in the circular RNA in the naming. For example, this is 2 alternative splicing circular RNAs. This one had 2 and 4, and this one had 2, 3, 4 exons, and also includes return introns, for example, or cryptic splicing site. I'll give you one example. So this particular gene locker is called FAM120A that can produce 5 different circular RNAs, but only 1 circular RNA that's produced from 2, 3, 4, 5 exons is functional, okay? So this particular circular RNA, look over here, localized to monoribosome, right over here. As a control, we see this translatable circular RNA, ZNF609, localized to polyribosome, and this circular PK3 is localized in the cytosol, is known as a microRNA sponge, so very different subcell localization. And this, so we're further knocking down this particular circular FAM120A with CRISPR-Cas13 technology. with 2 different get RNAs. Now we see the transition of its parental mRNA from polyribosome to monoribosome. As a result, we see this reduced protein translation by engaging with this translational inhibitor IGFBP2 of the sickle RNA. So this particular case will show that important to really define the sickle RNA you work with. All right, so the key question is, so many of them, so what do they do? So basically, there are different models, just like we know about link RNAs. They can do many different things with different modes of action. So they can work in the nucleus, they can work in the cytoplasm, or in the particular suborganelle regions. So I want to just to mention that for people who propose the sickle RNA work as non-coding decoys for microRNAs or proteins, please pay attention to their stoichiometric ratio to make sure the RNA you work with are sufficient enough to work as this model. And also, sickle RNA have been proposed to work as nucleotide scaffolds and also can work as competitors for mRNAs because that have the back-splicing junction site, making it very different from the linear RNAs. And in addition to these non-coding roles, there are also a few sickle RNA can serve as a translating templates, as a coding templates. So this is really a lot of colleagues' lab focus on sickle RNA over the last 10 years. Now have this picture. So in the rest of my talk, I want to focus on one of the roles that we found we think that is important for sickle RNAs and how from understanding the biogenesis of sickle RNA that allow us to appreciate their function involved in immunity, as well as how now we can apply the sickle RNA-based aptamer that can somehow benefit to PCAR-based inflammation. So one thing I want to ask, because the cells already produce the linear RNAs, why do they still need the sickle RNAs? So one hypothesis, maybe sickle RNA have different biogenesis from linear RNA that then have different functions. So I have talked about the cis elements, and then one identify trans factors because sickle RNA expression is a very cell and tissue specific pattern. If they all have the same cis element, they should have the same expression pattern, but the result is not. So here is a very useful plasmid we developed. So basically put this internal ribosome entry set with the start codon ATG and reverse with the TAA. We put this cassette into a flanking pair of complementary sequences, and then into a split EGLP gene, and put this entire cassette to the DOX-inducible PTET promoter. So as soon as we added this doxycycline, we see transcription of these transcripts and also both canonic splicing and back splicing that allows the formation of linear EGLP and cycloam cherry, allowing the translation of both green and the red color. So this also shows the efficient translation of sickle RNA of one of the quite early evidence. So using this cassette, now we focus on nascent sickle RNA to identify the trans factors that may affect the back splicing. So in this way, we identified more than 100 hits can specifically regulate back splicing. And quite unexpectedly, 16 of them have reported roles with viral and host immunity. So basically, somehow the sickle RNA biogenesis is limited upon viral infection. This basically is sickle RNA production at this stage upon viral infection is limited. So just remember this. And then we capture this nascent sickle RNA to compare the kinetics of back splicing. So it shows that compared to linear RNA, back splicing events is very slow, less than 1% of that of kinetic splicing. But some of the sickle RNA, not all of them, some of sickle RNA can accumulate to high levels. So I think that makes sense because generally speaking, sickle RNA are not, they are not accessible to linear RNA machinery, so making it more stable compared to linear RNA. So if the back splicing events is highly efficient, then our cells gonna full of sickle RNAs. So, so they're slowly produced, but they can be very stable. And also, we worked with sickle RNA chemical probing, and we found many of sickle RNAs, especially for those less than 500 nucleotides, they contain this imperfect upstream RNA region, okay, shown here. So that allows us to propose a model that circular RNA work as a group, okay? And I have mentioned that they are stable. However, they can be rapidly degraded upon certain pathological contexts, especially upon viral infection. We found the endonuclease called RNAs can rapidly and globally degrade circular RNAs. And here is half-life is on about 1 hour. So circular RNA is somehow its production is limited upon viral infection, and the steady state level circular RNAs are rapidly degraded. So we propose that the slowly production of circular and rapidly degradation can allow the circular as a group to modulate the homeostasis of immunity by interacting with PKR. I'm going to show you next slide about PKR. So normal situations, many circular they can keep this PKR as monomeric in active status. In fact, condition going to induce RNAs activation to trigger global circular degradation, release PKR for activity, for antiviral activity. And misregulation of this process is found in patients with autoimmune disease. So we observe the global reduction of circular expression and also this very subtle RNA activation and consistently augment the PKR activity. So PKR, so this is in the immune zoom, but this is what I understood about the immunity, at least it's kind of like 2018. So when viral infected cells, many of them can form the viral upstream RNA, trigger activation of RNAs. This viral and cellular mRNA degradation as part of antiviral activity. Now we know this pathway can also degrade circular RNAs. And this is RNA degradase, they can trigger those famous immune sensing receptors, leading to activation of interferon. In the meanwhile, those nucleic acid receptors with direct antiviral activity, including PKR, can also be activated by forming the dimers. So PKR contains two upstream RNA binding motifs and one kinase domain. Normally, in the inactive state, upon binding to long upstream RNA that can form the dimer and leading to activation of interferon and interferon-stimulated genes. And the meanwhile, it's known that short upstream RNAs within 16 to 33 base pair can block PKR activation, including the circular RNAs we identified. Now, if we got this primary cells from the patients, we put it into this ectopic expression vector I have shown you in the very beginning, that can make a lot of circular RNAs that can dampen PKR activation, dampen the PKR induced the EF2 alpha, interferon beta, and interferon beta trigger the gene expression. And those genes have been used as gene signatures for lupus diagnosis. So it's by 2019, it's basically for us in the lab by 2018, we will expect to see circular can do something that regulates immunity by studying their biogenesis. So that also suggests a potential of using circular as therapeutic strategy in pathological context with excessive PKR activation. So the question became, can we develop useful circular aptamers? If we want to do that, we wanted to make the circular in vitro. And those circular should have the short upstream region. And they should have little immunogenicity without modification. And we all know modification is so important to develop mRNA vaccines. And they should be more stable than linear RNA to work with. And also, can we see the similar thing in vivo? And is there any more robust activity to proof of concept that circular can really work as robust PKR inhibitors? So to address all of these questions, we have a team of 5 grad students together with postdocs. They work together we finally get something done. I cannot talk, sorry. So basically, long story short, we have tried to synthesize circular aptamers, including translatable circular RNAs by different strategies. So the conclusion was, it really depends on how you make them. So they can have the circular with different immunogenicity and yield. This is the 3 methods that we have shown here. One, just using T4 ligase that contain the short upstream duplexes, because the T4 ligase it does not really introduce too much additional nucleotides. So circular RNA made by this way, they still contain those desired upstream region, and they are very efficient PCAR inhibitors with more than 1,000-fold higher suppression than PCAR small molecules. Why? Because small molecule was designed to target the kinase domain, so it's not very specific. Very often, PCAR small molecule inhibitor can also inhibit like CDK kinase. And also circular made by this way has little immunogenicity and also very stable. However, circular made by permuted group 1 intron, they can introduce extraneous fragments, and those can distort circular folding and trigger immune responses. As a result, they are not very stable in cells. However, the good thing for the PAE method is the yield is high. So we really wanted to somehow optimize this catalytic intron method to make circular but without immunogenicity. So you may ask why the iron circles can trigger immunogenicity. At least in our experiment, this is initial PCAR activation assays. So when the purified PCAR incubated with 79 base pair upstream RNA, that can trigger PCAR activation. And however, incubation with those short upstream RNA has nothing to do with PCAR activation. Further incubate this circular RNA synthesized by T4 ligase can suppress PCAR activation, but not for those made by catalytic introns. Indeed, if we have this prolonged exposure time, we found those catalytic intron-made circular RNAs alone can trigger PCAR activation. That is because very often that catalytic intron can bring those extraneous sequences that can form this long upstream region. So to optimize this system, this some of the unpublished data, we basically has make a shorter extraneous sequences. To the bottom line is this 27 nucleotides long sequence that can form a stable stem loop with 5 unfilled base pairs that can barely interfering with the examined cargo sequences because it has very long, stable, and flexible single-strand sequences over here. And this is good to make the short upstream RNA like poly2A, that's circular aptamers, also can make a lot of those translatable circular RNAs, even up to, I didn't show the data here, to 7,000 nucleotides long. So now we basically using this optimized method. So this is the original junction site of the endogenous circular RNA. So in order to make a high yield, we have engineered this additional 27 nucleotide to this single body region without affecting this junction, without affecting the upstream RNA. So compared to our first version of the circular ligase, this newly produced circular RNA shows much lower suppression, much higher suppression on PCAR activation. So we have named this one as APIC-R, efficient PCAR inhibitor circular RNA. So here's the summary. You basically can get lots of circular RNA, 40 microgram circular RNA. That's sufficient for animal experiment and also structural analysis. To do this from 1 microgram DNA with 8 minutes co-transcriptional self-splicing with 2 steps of purification. So now we're able to make the circular RNAs barely induce expression of inflammatory cytokines in cells and in vivo. So is this work? we tried very hard to get this PCAR and the circular RNA using structure biology approach. However, it doesn't really work, still not work yet. We tend to use single molecule turf assay that can allow us to access RNA protein interaction dynamics. So we have labeled this cell-5 double RNA, linear 79 base pair, and linear 33 base pair double RNA, and double RNA within the circular RNA, all the circular RNA without double region in the turf surface, and further incubated with PCAR in green. This shows the results. So for the linear RNAs, doesn't matter 79 or 33 base pair, we always observe so-called transient binding events, okay? So on and off binding. So depending on the length of the double RNA, we see the PCAR have different retention on the molecule, but they has on and off. However, if we put this double region within the circular backbone, now we see stable binding events of this examined 120 minutes. As a control, circular without double region does not really bind to any PCAR. And then we have used this washing buffer to wash the additional PCAR from reassociation with this double region that gives rise to survival probability. Clearly shows here EPIC disc RNA shows much more stable binding and interaction with PCAR than linear double RNA. So this result shows circular RNA can form some distinct conformation to keep PCAR from the association upon binding, allowing this type of circular aptamer to robustly inhibit PCAR. So now we were able to make scalable disc RNA synthesis with little homogeneity and robust PCAR suppression. So how about any in vivo models? We all know lupus are very complicated. So we tend to use the psoriasis, which is a common chronic inflammatory skin disease, has the autoimmune and autoinflammatory features. And more importantly, as a molecular lab, it's easy for us to handle. So basically, we just shift the back skin of the mice and treat with the imicamab for 5 consecutive days. That gives rise to epidermal thickness at the phenotype. And in this model, we asked, is the RNA circular RNA PCAR axis dysregulated? The answer is yes. So we observed this transient RNA activation accompanying with global circular reduction and PCAR activation as a first wave. First with the PCAR followed by PCAR phosphorylation, lasted for more than 5 days. And we take advantage of this dynamic data. We did the RNA sequencing. It turned out that this PCAR cluster genes, not only PCAR, but also many other interferon double RNA sensor genes worked as the fast responding sensors in this imicamab model. Long story short, so if we knock out PCAR, we see this reduced fast responding inflammatory factors in this model. That suggests that PCAR is important, but previously overlooked axis in the pathogenesis of cirrhosis. Importantly, we not only see this in the mice, but also the activity PCAR cascade found in the lesional samples of cirrhosis patients. So how about the circular RNA overexpression? We decided to generate the first circular RNA overexpression mice. So we use the same cassette as I have just shown you in the very beginning that can strongly induce circular formation. We put this cassette into the classical ROSA 26 loci. And we did 2 different promoters, CG promoter, or EF1 alpha promoter. And in all examined tissues, we observed strikingly high circular expression, up to 1,000 copies per cell, or in certain tissue, but normally 400 to 600 copies. And this is a very interesting case because this circular RNA and the flanking intron is only human specific. It's not in mouse. So without this intronic pairing competition, so compared to what we see normally in human cells, Now we see strikingly highly induced the circRNA backsplicing, okay, over here. So that suggests our previous model about intrinsic pairing competition. Importantly, now we raised the mice for 1 year. We didn't see any problem during development. That suggests this additional supplement of circRNA is kind of safe, you know, unless you treat the animal challenge. For example, if we treat it with imocumab, and again, we see the reduced PCAR activation in the circRNA overexpression mice, and also reduce the expression of fast responding inflammatory factors, including M6, lots of the cirrhosis genes as well. All right, so now we have the genetic data supports important roles of PCAR and disc RNA in developing cirrhosis in mice. So can we achieve spleen-specific delivery of disc RNA that can benefit cirrhotic inflammation and phenotypes? So we have tried lots of different lipid nanoparticle delivery. This is a collaboration with Dan Pierce's lab at Tel Aviv University. So normally our MC encapsulated RNA is easy to be delivered into liver. So here we're able to deliver this circRNA into the spleen compared to liver, lung, and kidney. About 80% of the circRNAs are delivered to the spleen. And compared to linear RNA, circRNAs are a little more stable. I think about 6 to 10-fold higher, 24 hours later. And the good thing, not only for its stability, but also for its decay. So in the spleen, 48 hours later, 90% of circRNAs can be degraded. So they are stable, but they still can be degraded. So that suggests a very good PK in the future. So nevertheless, because we propose the circRNA work as the aptamer, we wanted to see whether the circRNA being encapsulated by the RNP are still structurable or folded in this correct conformation. So here is the result. So basically encapsulate with the RNP does not really change the circRNA conformation. So now with all of this fundamental understanding of the circRNA, we put this IV delivery to the mouse tail in the day 1.5, because we know the peak of RNAs activation started at the day 1 of MQ treatment. So 1.5 day of the imocumab treatment, we put this circRNA, and 1 day later, we sacrifice the animal and analyze the PCAR phosphorylation and also the epidermal thickness. And we were very happy to see about 40% of reduction of the epidermal thickness. So this is, we still don't know how exactly the splenocyte delivery can, alter or change the spleen, the skin phenotype so dramatically within 24 hours, but this is what we've seen consistently. So this is very encouraging data that suggests circRNAs with short upstream region can dampen excessive PCAR activation, at least in this model. So how about others? So can the upstream RNA aptamers benefit other pathological contexts? Another more severe context is Alzheimer's disease. I don't have to introduce more about the Alzheimer's disease, but here, starting from 2002, lots of indications and reports have shown the elevated PCAR and the PCAR phosphorylation with AD. Given the time limitation, I'm not going to share the data, but I can say that with the different adenovirus induced the circRNA delivery to neuron in the hippocampus, we're able to see the reduced neural inflammation mediated by PCAR, and also the rescued spatial learning and memory deficiency in both model of the AD, both FAD model and PS19 models. So this is how we want, beginning from the link arm processing and find they are slowly produced and can be fastly degraded upon viral infection. And they are very stable and they have very unique conformation, especially for those circRNA less than 500 nucleotides. And I want to mention that normally, normal size of circRNA is about 500 nucleotides, 500 nucleotides long. We found circRNAs containing this upstream region can dampen inflammation by PCAR, and more importantly, synthesized circRNAs can can be produced without immunogenicity. So that can open up a more broad application for the circular RNAs. And then we propose that this type of circular RNAs may represent a new type of RNA aptamers in the pathological context with unwanted PCAR activation, such as the lupus, psoriasis, and Alzheimer's disease. So this is only one application of potential circular RNAs because they are very stable, so they can be used the future potential biomarkers. And also, I have shown you insertion of internal ribosome entry site can driven circular translation. Actually, the first in vitro circular translation was published by Peter Sarro, now is professor at Stanford, in 1995. He studied the circular RNA translation a long time ago. But anyway, so now it's another wave of circular RNA mediated potential therapeutics because of their lack of immunogenicity, high stability, and can work as both aptamer mode and also translational mode. So this is the feature article published in the October 5th issue of Nature, talking about the RNA therapeutics next big thing is the rain. So fingers crossed. But I think this is a quite early field because only 10 years of real study of circular RNA since its rediscovery. Still lots of fundamental questions remain to be addressed, including trafficking, turnover, how the circular unfolding and function is related, how many of them have in vivo functions, how we can apply those fundamental understanding of circular RNA to design a better, more effective circular RNA-based new modalities. So we are doing some trafficking and turnover analysis with imaging in the lab. In the meanwhile, I'm thinking that this is a promising and also challenging field. For example, in the future, can we develop controllable durability of circular RNA cells? One thing is to understand circular degradation. Although they are stable, but they still can be degraded. So I'll show you two examples over here. This is a circular with very short stability. So we identify the motif on the circular that can be recognized by endonuclease. So if we remove those motifs, now we were able to have a higher or longer stability of the circular RNAs. In addition, this is a circular RNA, very stable circular RNA. So if we insert some core motif that can induce its degradation, okay? So hopefully in the future, the circular RNA-based therapeutics can be designed with a purpose. For example, if you want to develop a circular RNA-based vaccine, you basically don't have to express for a long time. But if you wanted to do some local circular RNA based protein replacement, you probably wanted to have the high stability of the circular RNA. So this is one example. And also we wanted to understand circular trafficking. So we show this is a very complex story. So that's why I didn't share with you now, it's under review. So we basically have shown that circular with some A motif prefer to retain in the nucleus in the stem cell. However, upon differentiation, those circular can be exported to the cytoplasm for function. So we also think in the future, a proper design of the insertion or exclusion of this A motif can also direct the circular RNA subcellular localization. So this is basically what we have been done about circular RNAs. It's a relatively new field, but promising in the future. I want to spend the rest of maybe 10 to 15 minutes to show a little bit of link arm processing, because I think messenger in another part also important for people who normally do not study non-coding RNAs. So what happened within the last 10 years about non-coding RNAs? If we look at non-coding annotation, more than 100,000 non-coding RNAs. However, functionally speaking, only hundreds of them have function supported by convincing experiments. So people have looked at their expression levels, interacting partners, folding conformation in cis or in trans, and then decided to what type of way to look at the functions using most appropriate loss of function or gain of function approach. But we still ask, are these analyses sufficient enough to appraise the link arm function? The answer is probably not. This here shows subcellular, an overview of subcellular localization of the link RNAs. It shows that link RNA works from all the way regulation of chromosome territory to post-translational modification. So that suggests that for the functional link RNA has localized to specific subcellular localization and then for function. Just like a functional protein, right? But unlike protein, RNA processing determines the subcellular localization, not this peptide. So quite a while ago, I have proposed the idea that if one wanted to really understand link RNA function, better to first look at their processing and the subcellular localization. Here is one example. So a long time ago, we want to study link RNA function in stem cells, both human ESL and mouse ESL. However, before we worked on this conserved link RNA, we first separated the RNAs either in the cytoplasm or nucleus for sequencing. The results are very interesting. For example, the mRNA is localized to the cytoplasm in both cell lines. However, in the mouse ESL, many of them link RNAs localized in the nucleus. However, in human ESLs, they have half of half, half nucleus, half cytoplasm. So we knocked down 5 pairs of the conserved link RNA at the time. We found only knockdown those in human ESLs can affect ESL pluripotency. However, knocking down the parallel link RNA in mouse ESL did not really show any detectable phenotypes. So what happened? It turned out that here is one example. This RNA called FAST, the FOXD3 antisense transcript. So in mouse ESL, this RNA is highly expressed, but once it's made, it's localized only in the nucleus. It's not processed by associating with splicing inhibitor PPIE, while it's also log link RNA in human ESLs bound to fewer splicing inhibitor can be fully processed and localized in the cytoplasm involved in the Wnt signaling transduction pathway. So this shows that conserved link RNAs may achieve functional divergence through non-conserved RNA processing, indicating the current model that we used to study link RNA, for example, mouse model, may not be sufficient to really access even conserved link RNA in humans. Even in the more bigger view, for people who study, because splicing is very cell type and species specific, if link RNAs also suffer from tissue or cell type specific splicing regulation, the outcome of this link RNA function can be very different. So far we still look at the bulk of cells versus subcellular localization pattern. So can we, look at the suborganelle level of localization that can allow us to access the stoichiometry between link RNA and protein as on-site. I mentioned at the very beginning that many of these link RNAs is localized in the nucleus. We found some of them conspicuously absent from patients with Prader-Willi syndrome, and the absence of such RNA can be used as diagnostic marker of Prader-Willi syndrome. And we also found this particular slurred RNA, which I'm going to talk about you today, localized in the nucleoli, and it's important to maintain nucleolar structure and function. And also some of other non-poly RNA, including NEAT1, which is key component of paraspicle. Paraspicle also plays an important role in immune responses. So all of these RNAs, they're very different, but they can modulate nuclear organization and function. I'm going to use this slide as an example to show you. So long story short, so this is actually also 10 years of work. We first identified SLERT a long time ago by look at our non-poly RNA sequencing data. So this RNA is kept by HAC RNA. SLERT, we named it SLERT because it can enhance pre-ribosomal RNA transcription. Here shows the gene structure of the pre-ribosomal RNA. And here is our this very famous nucleolars in the textbook. that have fibrillar center, dense fibrillar components that compose as a unit, which we call ribosomal DNA transcription and ribosomal RNA processing. And then the processed ribosomal RNA are assembled with protein to ribosome in the granular component over here, GC. So this is the same fibrillar staining in the textbook. So with 10 years now, this is what we see about the nucleolars. So many more FC-DFC units compared to what we learned before. So at least in my knowledge, like 7 years ago, I thought the per nucleolars only contain like 5, maximum to 10 FC-DFC units. But this is the HeLa cells that we normally see more than 100 of FC-DFC units. Even in human ES cells, more than 66 FC-DFC units, so lots of them. So SLERT, in the very beginning, we noticed that it localized to nucleoli and knock out this long-non-coding RNA, dramatically reduce ribosomal RNA transcription, and also leads to the reduced 28S and 18S RNA production. But we couldn't figure out how exactly this RNA work until we began to use single molecule FISH together with super resolution microscopy. So now we're able to dissect the SLERT mode of action as one FC-DFC unit. SLERT can promote POL1 transcription, but this RNA is never ever directly interact with POL1, but rather localized to its surrounding FC, called DFC region DDX21 protein. Normally in the cell, there are SLERTs that have sufficient room for the POL1 transcription. So bigger DDX21 ring, more pre-ribosomal RNA production. In the cells without SLERT, that made this ring much more smaller, that produced much fewer pre-ribosomal RNAs. So at that time, this paper was published, but we couldn't figure out deeper until we see ribosomal RNA and ribosomal DNA actually transcription only takes place between the border of DFC region and FC region right over here, and then have the radial flux nature going out. And SLERT interact with DDX21 in this way. In the presence of SLERT that can promote individual DDX21, which is an RNA helicase in the close conformation, therefore reduce the intermolecular interaction shown as here, resulting as a bigger DDX21 ring, and then sufficient room for POL1 transcription to occur. And in the absence of DDX21, that can keep this DDX, in the absence of SLERT, that can keep DDX21 in open conformation, leading to enhance the intermolecular interaction. As a result, we see this reduced DDX21 ring and FC-DFC unit, dependent POL1 transcription. So here is a single molecule turf again with Jiaqing Liu's lab at my institute. So we put this 14 kb long RNA on the turf surface and incubate with the DDX21 protein, always in the presence of 1% of the non-coding RNA, only 1%. And you see dramatic difference of the two. Okay, so DDX21 alone can hijacking this RNA. That's why we see dramatic reduction of RNA transcription within this dependent region. And this is in the presence of 1% of non-coding that can prevent this DDX21 to hijack RNA. So this is very important. Non-coding RNA regulates nucleolar structure and POL1 transcription. So when we approach nucleolars with this resolution, we had another question, because ribosomal RNA accounts for 85% of total RNAs. So how this multiple step error-prone radial flux pre-RNA processing and movement is achieved? So one hypothesis was this nucleolar protein and the RNA may have some compartmentalization in 3 dimensions that is highly coordinated with pre-RNA processing. I think I'm gonna skip this. Just wanted to show you that here is one new structure surrounding the classical DFC, which we call PDFC. This is what we think about the nucleolars now. So in the PDFC, so normally nucleolar is fixed separation protein rich the organelle. So it has lots of them are mobile protein. But in this PDFC, we observed two immobile protein. One of them is URB1, which is a huge protein, more than 2,200 amino acids. So without this protein, we see the dysregulated PDFC. So what happens of here? It turned out that this URB1 localized to the surface over here, providing a docking site for the 3' and the ETS to bind. This is the iClip data, specific band to over here. So with this binding that have correct conformation of rRNA, allowing the downstream SNP RNA to recognize and to process 28 siRNA. And without this URB1 protein, without this PDFC in both fish and mice, the animals have strikingly almost the lethal. I think in mouse it's lethal phenotype, but in fish it has a classical craniofacial phenotype. So and also in human, there are some disease situation with certain ETS pre-RNA. It's found in patients with Leber syndrome. So anyway, so just wanted to mention that nucleolars is quite complicated, and beyond the three classical layers, that there is another layer that can help the serum and pre-RNA binding and folding is essential for ribosomal RNA processing. Again, in this specialized nucleolar FCDFC unit, the RNA processing is also highly connected to its subcellular localization. So yeah, so this is advancing cell biology approach for linear study. So now we wanted to ask how these gentle RNA protein complexes are regulated and coordinated with gene expression, especially for some of these nuclear bodies have clearly related with these rare diseases. So in the future, I think it's really still need a higher resolution and the in vivo approach to understand the RNA biogenesis and function. We only worked a little bit on this. For example, able to label the RNA with different colors. We're also able to tracking the RNA in living fish embryo. So that shows the transcription memory. So here is de novo transcription, two alleles are very heterogeneity, and here the second cell cycle become very smart, synchronized. And this mRNP, just show you over here. I'm not sure whether it's working, but it's very cool. You can see the mRNP in the developing embryo. And we're also able to label 3 different colors at the same time. So for any of you who wanted to really look at the RNA, so the plasmids are available. So yeah, finally, thank you. So we have faculty position available for RNA biology at my institute. And this is my team, my collaborator, funding agencies. So really, if you happen to know anyone who will be on the job market, please refer to us. Thank you.